So it is recording. So for today, Heart 2. So Heart 1 was all about anatomy. And we do have a Heart 3 as well. That means Heart 2 and Heart 3 are all physiology. We're done with anatomy. Now we're going to learn about how the heart actually works. So we're going to describe the structural and functional properties of cardiac muscle and explain how it differs from skeletal muscle. So there's going to be a lot of review today. Briefly describe the events of the excitation, contraction, coupling in cardiac muscle cells. Describe and compare action potential in cardiac pacemaker and contractile cells. We'll name the components of the conduction system of the heart and trace the conduction pathway. And then we'll draw a diagram in lab of a normal ECG, name the individual waves and intervals, and indicate what each represents. And we'll name some abnormalities that can be detected on an ECG. All right, so a quick review on your PLA number 5. That's due today. So hopefully you did it already. So blood flow through the heart is going to be the superior and inferior vena cava plus the coronary sinus opening. Blood from all three of those sources is going to go to the right atrium, then pass the tricuspid valve to the right ventricle, pass the pulmonary semilunar valve to the pulmonary trunk, through both the right and left pulmonary arteries to each of the right and left lungs. And you'll notice that I went from blue to purple. Purple usually represents capillaries. That's where gas is going to be exchanged. And now we're oxygenated on this side. Our right and left pulmonary veins to the left atrium, through the bicuspid valve. Remember, we try before we buy. Left ventricle to the aortic semilunar valve, out the aorta, and then to the body. So on the new PowerPoint, there's a blank version that you can use for practice, and then the key as well. All right, so you should remember some key differences between skeletal muscle and cardiac muscle. This is all in your notes, but both skeletal and cardiac tissue are striated. And remember, they're striated because there are dark and light bandings, and the dark and light bandings are due to the actin and the myosin arrangement. Actin are thinner, so that's your lighter band, and then the myosin are thicker, and that's going to be your darker band. But no matter if we're talking about skeletal or cardiac, they're both are going to have that actin-myosin crossbridge, just like you talked about in BIOL 32. And the cardiac muscle structure and function or physiology is similar, but cardiac has some special, unique features that are key for its blood pumping role. So one of the big things is that skeletal muscle needs constant stimulation if you want to have a continued contraction. Versus cardiac muscle cells, they are autorhythmic, meaning they don't need any kind of stimulation. They go on their own. So if you guys remember the sarcomere, we have the all these different bands that you should have learned in BIOL 32. We have the H zone, or if you took Professor Newton, he calls it the bear zone, right in the middle, where you're only going to have the thick bands. The A band is going to be the thick and thin bandings. The I band on the outside are just going to be the myosin actin, the thin one only. And then remember, every sarcomere goes from Z line to Z line, or Z disc to Z disc. And the sarcomere is the smallest functional unit of muscle. So one thing about skeletal muscle is that they have to use a motor unit. So skeletal muscle fibers contract as a function of motor units. And if you remember from B32, a motor unit is one motor neuron and the fibers it controls. If we're talking about muscles that need really precise movements, a motor unit is only going to control a few fibers, so like the muscles in your eye or maybe the muscles in your fingers. But anything for like really bulk movement, maybe like your back muscles or your leg muscles, those motor units are going to be controlling many fibers at one time. And then a little bit about neuromuscular junction physiology. So please review this if you have not yet. No need to write this down. This is just review. If you need to go back and review, go back and review. But if you remember from B32, an action potential arrives at a motor neuron that action potential, that positive 30, is going to cause calcium channels, voltage-gated calcium channels in the axon terminal to open. The calcium is going to come into the axon terminal. That influx of calcium is going to cause the vesicles with the acetylcholine to go to the membrane to be released by exocytosis. Once the acetylcholine is in the synaptic cleft, then it's going to diffuse over to ligand-gated channels. Those ligand-gated channels are also sodium channels. Those are going to open up as soon as acetylcholine binds. That's going to allow sodium to leak into the skeletal muscle cell, and that's going to bring us closer and closer to threshold until we reach it. Once you reach threshold, then that's going to switch us to voltage-gated channels, and those voltage-gated channels are going to stimulate the entire skeletal muscle fiber that we're talking about. And then as soon as that calcium reaches the T-tubules and goes deep into the skeletal muscle fiber, that's going to open another set of voltage-gated calcium channels that are going to allow calcium to come out of the sarcoplasmic reticulum and bind to something called tropomyosin. It depends on which professor you took, but it binds to the troponin. Calcium binds to the troponin, which moves tropomyosin out of the way, which allows for the actin and myosin to come together and do the cross bridge. All of that. You guys remember all those steps you guys learned in B32. Again, if you need to review that, make sure that you do. But with the skeletal muscle, again, you need that motor neuron. You need the acetylcholine to bind to the ligand-gated channels to get us to reach that threshold. Cardiac muscle cells are completely different. The heart is what we call a functional syncytium, which means that individual cells work with neighboring cells for a coordinated action or contraction. So they all are working together. Okay, versus skeletal muscle, they're all working separately from each other. They may all work at the same time so that you can lift something heavier, okay, but they're all working individually. The heart works together as a whole. And so the heart is filled with a couple set of cells, but one of those are going to be autorhythmic cells. Autorhythmic cells contract without neural or hormonal stimulation. So they can reach threshold all on their own. They allow for that action potential to be sent all on their own. So questions about this so far? Okay. So just like with skeletal muscles, okay, the calcium binds to the troponin, which moves that other protein, which is called tropomyosin, out of the way. That exposes the active site on actin, which allows myosin to connect and create that cross bridge. The thing about cardiac muscle cells, though, is that troponin should stay inside of the cardiac muscle cell at all times. It should stay inside. And so if you have a positive blood test for troponin, that indicates that a patient has had cardiac muscle cell damage and that troponin was allowed to leak out into the blood. Okay, so this could mean a myocardial infarction or an MI or several other cardiomyopathies, which are other conditions of the heart. And this can help differentiate between an MI and angina. Angina is when you are experiencing coronary ischemia, so your cardiac muscle cells are hungry for oxygen, but they haven't been damaged yet. They're not dead yet. Okay? You only have dead cardiac cells or damaged cardiac cells with an MI, not with angina. So this can help you differentiate between angina or chest pain from angina or a myocardial infarction. And so if the level goes down over a few days, it means the damage has been halted, okay, and there's no longer cardiac muscle cell death going on. But after a few days, if troponin is elevated, then damage to the heart is continuing, and so you're still experiencing that ischemia. Those muscle cells are still starving for oxygen and are dying. Okay. And all that should be in your lecture notes. So any questions about blood troponin? Okay. So then a little review. So about the differences between skeletal and cardiac muscle. So intercalated discs, skeletal muscle cells do not have it, but cardiac muscle cells do. Branching, Skeletal muscle cells do not have branching. So if you guys remember looking at it underneath the microscope, they are cylinders of individual muscle cells that do not connect, versus cardiac muscle cells have all this branching so that they are all connected to each other. Skeletal muscle cells do not have autorhythmic cells, so they need motor neuron stimulus from the motor unit. Cardiac muscle cells, though, are autorhythmic, and they do have autorhythmic cells. Skeletal muscle cells do not contract as a unit. Hey, they are all working individually. Again, they can work at the same time, but they're not necessarily working together. And then cardiac muscle cells, yes, they do contract as a unit. They're all working together. The calcium source for skeletal muscle cells is the sarcoplasmic reticulum. But then cardiac muscle cells actually have a second source. They have the sarcoplasmic reticulum, and then they have calcium channels in the membrane that we'll talk about later. And then the ATP source, skeletal muscle cells, you should know, can do both aerobic and anaerobic pathways. You use aerobic more for like long distance running, or maybe like yoga or things like that. Okay, anaerobic you would do for things like HIIT, which you're doing like really intensive movements for a short amount of time, or really heavy lifting. So if you go to the gym and you're trying to reach your maximum and you're only doing like 2 to 3 repetitions, you would be using anaerobic then. Okay, but cardiac muscle cells have to use aerobic all the time, because remember, they are working 24 hours a day, 7 days a week, 365 days a year, so they need that constant energy. They cannot live off of 2 ATP per glucose. With anaerobic, they have to do aerobic to get that 32 ATP per glucose. So any questions about the difference? This should be review for you. Okay. Anybody still writing down the table stuff? Okay. So again, remember, skeletal muscle fibers do not have any branching. Here's an individual skeletal muscle cell. Here's another individual skeletal muscle cell, another. They are parallel to each other, but they do not cross over. They do not connect to each other. Versus cardiac muscle cells, they branch in different directions. Okay, so they are all interconnected. And you can see the intercalated disc. I'll do it right next to the dark pink lines, so I'm not covering it. But all of these are intercalated discs. Okay. We're not going to focus on smooth today. We're really only looking at the differences between skeletal and cardiac muscle. But if you remember smooth, they do not have striations. Their cell shape is like, not like that. They have a spindle-shaped cell. And so those intercalated discs, they are made from gap junctions. So intercalated discs are gap junctions, which are these cellular junctions that have pores. And those pores allow for the ions to travel from cell to cell, so that when we depolarize one cell, then we can depolarize the next, and the next, and the next, to send those action potentials. And then also desmosomes. And then desmosomes are the anchoring junctions that have anchoring proteins on the inside of the cell, and then they're connected by fibers. These anchoring proteins, they allow those cells to stay together, because the heart is contracting really forcefully. And so you don't want those connections to come apart. So we have the desmosomes with the anchoring proteins on the inside of each cell, so that those cells are not coming apart from each other whenever the heart contracts. Okay. And then remember, they do have that branching, so that they're all connected, so that they can allow for the ions to move through these pores and the gap junctions. And this allows the action potential to get sent from cell to cell to create a wave-like contraction of the heart. But that should also be in your notes. So questions about the intercalated discs? Okay. So you may have questions about this either on the written or on a practical, but we won't do any microscopy with the cardiac muscle cells this semester. So you don't have to look at them under a microscope or identify anything under a microscope for this section this semester. You were already tested on that in B32. So, and then skeletal muscle, again, the motor units contract independently. So with skeletal muscle cells, they must have a motor neuron for stimulation or contraction. They cannot work without having that acetylcholine release. So with our cardiac muscle, cardiac muscle in general, the heart has two types of cells. And I think this is already your notes too. It is? Let's see how much it's in your notes. Yeah, actually all of this is in there. Okay. So we have two types of cells. And for those of you who are not following along with your notes right now, we have contractile. And I'm going to put CMC for cardiac muscle cells because I'm running out of room. And then we have pacemaker cells. And these are non-contractile. And so the pacemaker cells, they depolarize spontaneously. They don't need any stimulation. So that depolarization or that leaking in of sodium to reach threshold, that's happening automatically. Again, they are autorhythmic. And this is allowed to happen by those gap junctions. Once a little bit of sodium leaks in, then you're going to leak into the next cell, and then the next cell, and the next cell, and they're all going to get excited at the same time. And so there's no neural input necessary, which is why you can cut the nerves and transplant a heart, and because a heart will work on its own. Now, like we said before, cardiac muscle cells all work together while skeletal muscle cells work independently. So if we wanted to have a stronger contraction and we needed to have more skeletal muscle cells to do it, does anybody remember what that's called? When we call on more skeletal muscle cells and more skeletal muscle cells, if we wanted to have a,
Synergistic.
Not synergism, it starts with an R. It's called recruitment. So if I wanted to pick up something really light, like my case here, okay, it would only take a few of my hand skeletal muscle fibers to do that. But if I need to pick up something heavier, I would need to recruit more skeletal muscle fibers in order to allow for that larger lift, for that heavier lift. Cardiac muscle cells do not require recruitment because they're all going to work together. It doesn't matter if we're sitting down resting or if we're sprinting. Okay, it does not matter. All those cells are working together the entire time. Versus skeletal muscle cells, it just depends on how much force you need is going to depend on, or it's going to determine how many skeletal muscle fibers are recruited. Okay, so that's different between our cardiac and our skeletal muscle cells. So cardiac muscle cells do not require recruitment. They're all going to work together no matter what. So questions about those, well, and then with these two different cell types, pacemaker cells, just like the name says, they're going to set the pace of the heart. So they're really only to excite the rest of the heart versus the contractile muscle cells. Those are the ones actually doing the contracting and doing the work of filling and ejecting the blood out of the heart. Okay, so we have a subset of cells just for setting the pace and the timing. And then we have the other set of cells that are for doing all of the work, okay, for contracting to allow for the filling and ejecting of blood. So where do we say is the calcium source for skeletal muscle cells? Sarcoplasmic reticulum. The sarcoplasmic reticulum. And the sarcoplasmic reticulum for skeletal muscle cells is going to release all of the calcium that's needed for contraction. Okay, so any calcium that we need for contraction with skeletal muscle fibers is all going to come from that sarcoplasmic reticulum. And you do have like a little table at the bottom of your lecture notes about calcium source, ATP source, and the size of their mitochondria. We're talking about that now. So there are two sources of calcium for cardiac muscle cells. We have slow calcium, and calcium is 2+, calcium channels, WI for within the plasma membrane. And then we have what are called calcium sensitive channels. Within the sarcoplasmic reticulum. And what's unique about these two sets of channels is that the slow channels, this allows for 10 to 20% of the calcium needed for contraction. And this one is going to work first. Okay, so we have these slow calcium channels that are going to allow calcium to leak into the cardiac muscle cell, only 10 to 20% of the calcium needed for contraction. And then these channels over here are calcium sensitive. So they detect when the calcium enters the cell. And so once that calcium enters the cell, then these channels get activated. They work a lot faster, and they're going to release the remaining 80 to 90% of calcium that's needed for contraction. So these calcium sensitive channels are going to open in response to the slow calcium channels in the membrane. So questions about those two channels and how they work. Okay. And they are both voltage gated. All of the cardiac muscle channels that we're going to talk about, they are all voltage gated. Okay. Anybody still writing this stuff down? Okay, I'll wait a second. Do you have a session on Thursday? Mm-hmm. How'd it go?
Well, I had 3 people.
Okay.
Yeah.
Everybody good here now? Okay. So ATP source, I'm going to write this down. For skeletal muscle, and this is in your table on your lecture notes. But for skeletal muscle, they use anaerobic and aerobic respiration versus cardiac muscle uses aerobic only. Ooh, too many A's. Now, in your lecture notes, it already says that these skeletal muscle mitochondria are of average size. Bless you. They're average size mitochondria, and they only take about 2% of the cell volume of our skeletal muscle cells. Versus cardiac muscle, and rightfully so because they never stop working, these cells, they have large mitochondria. And these mitochondria take up 25 to 35% of the cell volume. So because they not only do they have large mitochondria, but they have so much of it, so many copies of it, that it takes up 25 to 35% of their cell volume, cardiac muscle cells are highly resistant to fatigue. Can you imagine our heart getting as tired or as quickly as our skeletal muscle cells? That would be horrible. Those stairs in the SC building would take me out every time. Dr. B is texting me. Okay, everybody have these notes written down. Okay. So our heart is made up of pacemaker cells, which are what we're going to talk about next. We're going to talk about this entire conduction pathway. We use a conduction pathway because our first set of pacemaker cells start up here at the right atrium, but it's not only the right atrium that needs to get excited and contract. We need to be able to contract the top of the heart all the way down to the apex of the heart. So there's a whole conduction system that allows us to excite the entire heart and in a timely manner. Because the whole heart is not going to contract at the very same time. The atria have to contract first to fill the ventricles, and then the ventricles contract to eject the blood. Now this is happening like within milliseconds. Hey, but they are contracting on a little bit of a different time. So this is a review of the regular action potential from our skeletal muscle cells. So for skeletal muscle cells, after acetylcholine binds, hey, that opens up these ligand-gated sodium channels and sodium leaks in. So we're going to get closer and closer to our to our threshold. So if we start at negative 70, we go from negative 70 to negative 69, 68, 67, 66, and then 65 is our threshold. If for other professors, it may have told you negative 55. That's not really super important because depending on the cell, there's going to be a range for threshold. It could be at negative 65. It could be at negative 55. They're all going to be different for different cell types. Okay. So once we reach that threshold, whatever that number is for that cell, then we're going to switch to voltage-gated channels. And then we have our depolarization phase. This is our rapid depolarization. So voltage-gated sodium channels open, and again, we have a rapid influx of sodium. And remember when we were talking about those fluid compartments, I reminded you that we have lots of sodium on the outside of the cells to begin and a little bit on the inside, so that when this depolarization happens, we have this huge and rapid influx of sodium because of that unequal concentration. The greater the difference in concentration from the inside to the outside, the faster that movement can happen. Okay, so we have this rapid influx of sodium into the skeletal muscle cells. Then once we reach our max of our action potential at about positive 30, then we have the repolarization phase. Okay, we're going to switch from voltage-gated sodium channels to voltage-gated potassium channels. And in this case, the concentrations are flipped. We have a lots of potassium on the inside, less on the outside, and that causes an efflux of potassium out of the cell. Now, once we get back to around our resting membrane potential, then we're going to have what's called the refractory period. Come on. Goodness gracious. There it goes. Then we have our refractory period. And during this time, we cannot refire any action potentials. Okay, no more stimulation can occur until the next one. And during this time, we are going to reestablish that membrane potential. Because even though we're at negative 70 again, this negative 70 looks way different than this negative 70 because we just moved the ions everywhere. We made the sodium rush into the cell. We made the potassium rush out of the cell. So those concentrations are now different. So this refractory period allows us to reset back to these concentrations. Does that make sense? So when we send it again, we're set up for the same thing. An influx and then an efflux. Now remember, this is all done by a sodium-potassium pump. The resetting of the action potential is done by a sodium-potassium pump, and that requires active transport. And what does active transport require? ATP. So any questions about this review? Who's still writing this stuff down? And I'll wait. I have coffee to drink it. Let me know if my head's in the way. I'm about the way. You had fun studying acupuncture? I don't know if they'll tell you the same thing. It's easy to have fun with it when you're done with the class. Which is why I have so much fun with it too.
Yeah.
Okay, everybody good now? Okay. So now I hate to tell you this, after you learn the acupuncture this way, the cardiac muscle cells are going to be a little bit different. Okay, but some things are going to be the same. So we still have that threshold. I'm not going to tell you the values. If you want to learn that on your own, that's fine. I'm not going to test you on the values, the millivolts. You don't need to know what threshold is, what our resting member potential is. Okay. If you want to learn that, you can. It's like negative 60, negative 40 is the threshold, and then between 0 and 10 is the peak. But again, I'm not going to test you on that. So the depolarization that happens before threshold is still like the action potential, where we have sodium leaking in. Okay? But again, this does not require acetylcholine. This is all happening autorhythmically. It's all happening spontaneously. So we have the slow leaking in of sodium until we reach threshold. And what's different with cardiac muscle cells is that for the rapid depolarization that happens next, it's not sodium, it's actually calcium. So after we reach threshold, we switch to voltage-gated calcium channels, and those calcium ions are going to come in. It's still an influx on this side, so ions are still moving into the cell, but instead of it being sodium, now it's calcium. Okay? So calcium rushes into the cell to get us to the peak of our action potential. And then the repolarization is the same. We switch to voltage-gated potassium channels. Potassium moves out of the cell. We have an efflux of potassium out of the cell until we go back down to our original resting memory potential. And then it happens again. So once we reach our resting memory potential again, the potassium channels close, and then we have slow voltage-gated sodium channels open to help us reach threshold once again. And the rate that the sodium comes in right here during this phase is slow leaking in. The rate of that is going to determine how fast our heart beats. So that rate serves as our timer. Ooh, go back. So if we're looking at these action potentials and we were to speed up the rate of the heart, we would just have more of these closer together. So it's a little bit different. And again, this whole thing that we're talking about here and what we're going to be talking about next is our intrinsic cardiac conduction system. And we'll use the word intrinsic a lot this semester. Intrinsic means that the regulation is happening within that organ itself. Okay? It doesn't need input from other parts of the body or the nervous system. For this, it's all intrinsic. It's all happening within the heart with no outside influence. And so in your notes, I mentioned that all ions are voltage gated. Like I said, all ion channels are voltage gated. And then it's all or nothing, kind of like skeletal muscle cells. All cardiac cells are going to contract or none of them do. Right? So they all have to work together. All cells are connected by a gap junction, except for the neighboring cells between the atria and the ventricles. Now, I'm not a great artist, so I'm not going to pretend like I could draw an anatomical heart in 10 seconds. So right atrium, super simple. Right ventricle, left atrium, left ventricle. So all of these cells are going to work together and have gap junctions that connect them, but they are not connected to the ventricular cells, okay? So the atrial cells and the ventricular cells are not connected by gap junctions. There are no gap junctions here. And the reason for that is because they contract in different directions, okay? The atria contract downward in order to push that blood from the atrium into the ventricles. Okay, so they need to contract down to fill up the ventricles. And then the ventricles need to contract upward in order to get that blood up and out, either past the pulmonary semilunar valve or up and out and past the aortic semilunar valve. Does that make sense? Okay, so they cannot be connected by gap junctions because they're contracting in different directions. We need the atria to contract downward first. So the atria are going to fill up. They're going to contract to push that blood down into the ventricles. And then once that happens and we fill up the ventricles, then the ventricles are going to contract upward in order to push the blood out of the heart and eject it. Okay? So they are not connected by gap junctions. And the timing is different. The atria contract and then the ventricles contract. And then the atria and then the ventricles. Yeah.
So when the atriums and the ventricles are pumping, is it the right atrium and left atrium?
Yeah, they're both working at the same time.
Okay.
So they're both contracting downward while the ventricles are both contracting upward.
In this question, we have to answer the question like they are contracting in a different direction.
Yeah. So that question in the lecture notes where it says, where's that at? Oh, except neighboring cells between atrium, ventricles, and Y. Because one, they contract at different times. Atria contract first, and then the ventricles contract. We have to fill, and then we have to eject. And because they're contracting in different directions. Atria contract down, ventricles contract up. Okay, any quick questions about the action potential of the cardiac muscle cells? Anybody still writing notes down from this slide? Okay. Oh. Okay, so this is just what the image from your textbook. So this first stage right here is called the pacemaker potential. I'm not going to make you name it specifically that, but this is the slow depolarization due to both the opening of sodium channels and the closing of potassium channels. And notice that the membrane potential is never a flat line. When we looked at the cardiac muscle cells, there is a flat line where we have no contraction or no stimulation happening at all. Okay, once I'm done picking up my pencil, there's no more stimulation. Okay, versus a cardiac muscle cell, that stimulation never stops. Okay, those heart muscles are always working, always contracting. They're always excited. And so there's never going to be a flat line. That's a bad thing. We don't want a flat line. Okay, and then after that, stage two is the depolarization phase. This is the rapid. The action potential begins when the pacemaker potential reaches threshold, and the depolarization is due to calcium now influxing through calcium channels, and this is a rapid influx. And then we have the repolarization phase going down this way, and that's going to be calcium channels now closed, potassium channels open. This allows for an efflux. So we have the leaving of these positive ions. It's going to bring our membrane potential back down to where we started. Questions about that? Okay. That is a pacemaker cell's action potential. Remember we said we had two types of cells. We have pacemaker cells, and then we have contractile cells. And the contractile cells have a different action potential. And so with pacemaker cells, the depolarization phase happens very, very fast. You can see that there's a very small window here, and that incline is so steep. Okay, when it's more like this is taking more time. No, this is happening extremely fast, okay, to get to our max of our action potential. And that's only due to sodium channels, okay? And once we get to the peak of our action potential here for contractile cardiac muscle cells, at this point, there's a phase called a plateau, okay? This plateau is going to allow our cardiac muscle cells to contract for longer. So a action potential for skeletal muscle cells is only about 1 to 2 milliseconds long. Very, very fast, okay? Cardiac muscle cells are fast too, but the action potential lasts up to 200 milliseconds. So 100 to 200 times longer of an action potential in our cardiac muscle cells versus our skeletal muscle cells. So this allows us to fully fill our ventricles, and also allows us to eject that blood and eject enough of that blood out, okay? So these sodium channels open, they open fast, and we have this rapid increase, very steep increase in our membrane potential. So the membrane potential is going to go all the way up to our peak here. And then when we get to the plateau phase, during this phase, sodium or potassium channels are still closed. We're not in the repolarization phase yet. We have slow calcium channels open, and the sodium channels are going to be inactivated. So these ones close down over here. And we have slow calcium channels open that allow us to keep that high membrane potential without it fully falling down. Okay, and because why we need this and why we need those slow calcium channels to be open is because as long as calcium is available, no matter in what concentration, so as long as calcium is available, cardiac muscle cells can contract. So we have this plateau phase where we have slow calcium channels open, and it kind of keeps this action potential or this membrane potential high for a little bit. So these slow calcium channels, these allow for a longer contraction because we're keeping calcium available. And a longer contraction means proper ejection, which means that we can eject as much blood as possible to get it out to the body. And because we have this plateau, and because the action potential is lasting longer, I'll show you the next slide, and then I'm going to come back. Because the action potential is longer, the refractory period is also longer. So this means there's a longer amount of time where not another action potential can be sent. Okay? And you can see after repolarization, you have this big space here of time. That time allows for the ventricles to properly fill before we send another action potential. Okay? So again, that plateau keeps calcium available so that we can eject enough blood, but then it makes us have a longer refractory period, so we have more rest time in between, so that the ventricles can fill properly before the next ejection. So this plateau allows for a longer contraction, which means proper ejection, and a longer refractory period, which means proper filling before the next action potential. And then just like other action potentials, then you guys actually, before we do that, you guys write that stuff down and then I'll show you guys the next part. So I can erase it. Okay, did you guys write that little bit of notes right there? Anybody still need it up? Okay. So I can show you the rest of the action potential. So then during our repolarization phase, calcium channels are now closed, potassium channels open. We have an efflux, which brings us down just like with the other action potentials, and the resting potential is restored. And during this time, calcium is pumped back into the sarcoplasmic reticulum so we can have that rest period in between and give the ventricles time to be filled up before the next action potential. So I will say, just to kind of ease your stress, it's important to understand this, especially when we talk about the filling and ejecting of the heart in Heart 3. We're going to talk about the cardiac cycle. So it's really important to understand what's happening here. So if you need a review, go back and review. But for testing purposes, I'm only going to test you on the pacemaker action potential. That does not mean don't study the other one, because again, you need to understand what's happening in order to understand the cardiac cycle. But I'm going to give you a little bit of relief and tell you don't have to stress over memorizing the action potential of the contractile muscle cells. You're welcome. Focus on this action potential, okay? Okay, any questions about those action potentials?
Can you go back?
Yeah, to this guy right here?
Yeah.
Okay. I'm always a little weary about telling students that I'm not going to test them on something, but study it, review it. Again, it has implications for the next lecture. It's especially true in Bio 33 that everything builds on each other. So this is not a class that you can get away with just memorizing your way through. You have to really understand it. If you can explain it to somebody else, then you're at a pretty good spot. So that's the goal you guys should all be aiming for when you're studying, is can you explain it to somebody else? And it doesn't even have to be another scientific person or somebody else taking biology. Can you speak about it out loud and make sense of it? Okay, so I challenge you all to do that with all of these topics here, because when you talk about a topic in B33 out loud, so let's say you are talking about the action potential here. When you talk about it out loud, put all your notes away, everything away. Well, study it first so you know it enough. Then you put all of your notes away, and then you start to talk about it out loud. When you talk about it out loud with your notes put away, you get to a spot where you're like, oh, I don't know what happens next, or I don't know why this happens. I'm not explaining it well enough. Go back, study some more, and then try it again. Okay? And again, it doesn't have to be to somebody who is biologically in tune, okay? It could be to a stuffed animal. It does not matter, okay? Just, you need to talk about it out loud. Okay? It's good to think, yes, but it's going to be much more effective if you can talk about it out loud.
So for the cardiac, or local depolarization, it's not sodium, it's calcium?
No, for the local, and it's not even really local because we don't need a ligand-gated channel, but the slow leaking in, that's sodium still. So we still need sodium to reach threshold, but once we reach threshold, then we switch to calcium channels for the pacemaker cells. For the contractile cells, it's all sodium for the depolarization.
And for the rapid depolarization?
Now, for our contractile cells, there is no rapid and slow. It's all rapid. All of this is happening within a couple milliseconds. Very, very fast, and all it needs is sodium.
Sodium, okay.
But for these ones, we start off with sodium slow. Once we reach threshold, then we do the rapid, but it's calcium instead of sodium. Yes?
For the slow sodium, like within channels, is that also voltage gated, but it's just the sodium?
Yeah, it's still voltage gated. Yeah. They're all voltage gated. But it just happens spontaneously rather than needing a messenger to tell it when to do so. Any other questions? Okay. So next we're going to talk about something called the sequence of excitation. So we said before, the atria contract first and then the ventricle. So there has to be a pathway to go from the top of the heart down to the bottom of the heart and to get the ventricles to contract. So what I'm going to describe to you here is going to be our sets of pacemaker cells throughout the heart that are communicating with each other. Our first set of pacemaker cells is going to be in an area called the SA node. Okay, it stands for the sinoatrial node. Now I said only hormones, you can use abbreviations. You can use abbreviations here too, but just for this sequence. Okay, you don't have to say sinoatrial, you can say SA node. So we have the SA node. This is the true pacemaker of the heart. And it depolarizes about 75 times per minute. Now, one thing you have to pay attention to is that it says it depolarizes 75 times per minute. Now, that does mean that the heart is going to beat about 75 beats per minute, but that's a different measurement. Right now, we're just talking about excitation and depolarizing the cell. So if I ask you for a depolarization rate, you're going to tell me times per minute. If I say the depolarization equates to what heart rate, then you would tell me beats per minute. Okay, those are two different units and two different measurements. So depolarization rate is in times per minute versus a heart rate is in beats per minute. Okay, does that make sense? Because right now we're only talking about the excitation. We're not talking about the contraction just yet. So the SA node is a pacemaker of the heart. It depolarizes about 75 times per minute. Next set of pacemaker cells are at the atrioventricular node, or the AV node. This is going to be closer to that connection between the atria and the ventricles. Now, from the SA node to the AV node, there is an impulse delay of about 0.1 seconds. Why that's important is because we said the atria have to contract first and then the ventricles contract after that. So we need that impulse delay so that the timing is right. So the atria can contract and fill up the ventricles, and then after that, the ventricles can contract and eject that blood. So there needs to be a delay so that you have separate contractions happening at different times. Okay, does that make sense? Now, this AV node, if the SA node were to be non-functional, let's say your SA node is damaged, does not work, then your AV node could take over the pacemaking of the heart, but it does it at a slower rate. So if no signal from the SA node, then it's going to depolarize at about 50 times per minute, which is enough to keep you alive, but you'll feel a difference. Right? If your SA node isn't working and you're only depolarizing 50 times per minute, which means that your heart is only beating about 50 beats per minute, for an average person who's not super athletic, you're going to feel the difference in that. You have a difference in oxygen delivery, a different in nutrient delivery, and you're going to feel the difference. After a while, you may need a pacemaker to correct that, or you maybe need to put on medication to increase your heart rate when that happens. They'll probably do medications first, see if that works, and then after that, if that does not work, then they may need to put you on a pacemaker.
So this one is also 15 minutes per second?
Say it again?
This one is 15 minutes per second?
Yeah, so 50 times per minute, which would equate to about 50 beats per minute. The average heart rate is around 75 beats per minute, resting heart rate. But anything between 60 and 100 is considered normal. Now, both of these set of pacemaker cells are located in the right atrium. Next is what we call the AV bundle. If you're looking at an older textbook or an older reference, sometimes it's called the bundle of His. But we are, as a scientific community, moving away from naming things after people, men, hey? Because sometimes it's not the men who do it, who discover it, and then we find out later it was a woman who contributed, but whatever. We're moving away from naming things after people, and we're giving them more scientific names. So this is the AV bundle, but it may still be referred to as the bundle of His. But the AV bundle, this is located in the interventricular septum. But this is not pacemaker cells here. This is just a connection between the atria and the ventricles. So remember we said that there's no gap junctions between the cells of the atria and the cells of the ventricles? Well, this is how they communicate with each other without those gap junctions. So this is the only connection between the atria and ventricles. Because remember, there are no gap junctions between those two chambers. The next part in the excitation sequence are the bundle branches. We have right and left bundle branches. This is to excite the right and the left ventricles. Okay, but this is also not a set of pacemaker cells. This is just a connection to the right and to the left ventricles. And this goes down the septum towards the apex. And then our last set of pacemaker cells are now called the subendocardial conducting network, but you may still have sources that call them the Purkinje fibers. I'm okay with the Purkinje fibers. I still use Purkinje fibers, and that's just because it's way easier than the subendocardial conducting network. Huh? I mean, I guess Gordon says I'll give you an extra quarter of a point if you can say the subendocardial conducting network instead of Purkinje fibers. Okay, these are located, these cells, and these are pacemaker cells as well, they are located at the apex and sides of the ventricles. And so if there's no signal from either of the SA and AV node, let's say both of those nodes are non-functional, or maybe they're functional, but there's something wrong with the AV bundle. There's something wrong with the AV bundle and there's no connection between the atria and the ventricles. So if there's no signal being received by the ventricles, then the Purkinje fibers can depolarize at an even slower rate than the AV bundle. They depolarize at about 30 times per minute. And you cannot live at 30 times per minute. So it's important to know that the SA node is a true pacemaker of the heart. It depolarizes 75 times per minute. If that's not working, the AV node can pick it up, but it's only going to depolarize 50 times per minute. If neither of those are functional, or if there's something wrong with the connection, The Purkinje fibers can excite the ventricles, but that's only going to happen at 30 times per minute. Okay, so every time you go down that sequence of excitation, the depolarization rate gets slower. So questions about that? Yeah.
So pacemakers, those are just the first two?
First two and the last one. The ones that can depolarize. Yes.
I know somebody that had an issue with the no signal from the SA nodes and the AV nodes.
Uh-huh.
And he has a pacemaker and a defibrillator. What is the defibrillator do in that instance?
It can reset the heart if needed to kind of get it to restart again.
They have to replace it, the pacemaker, every.
Like, it depends. If it has leads, it's usually about 5 years. But if it's a leadless or wireless one, it's about every 12 years. So it depends on the kind of device that you have.
And the defibrillators, like every 10 years?
I think so. I'm not sure about the defibrillators.
My brother has one of them.
About 10
We
years?
call him Iron Man.
Iron Man.
And can it be repositioned anywhere in the heart, or it will be stick on a one position?
I believe they put it in the same position, but again, I'm not a cardiac surgeon, so I couldn't tell
He
you.
has his right in the center.
Right in the center?
Yeah.
Any other questions? Okay, so I want you guys to take out your lab really quickly and we can do one of the activities together and get it out of the way today. And it's basically everything we just talked about.
So we have to like observe something.
Page one.
Oh, okay. Oh, it's on page 1. I don't know why they number it like that.
It's 1, 2, 3, 1.
Yeah, every lab starts off again at page 1. So it's whatever the lab is after the heart anatomy lab. I think because before they put it together in a manual, they all used to be separate sheets and nobody's ever. And I actually did renumber it. And I sent it to the one who makes the order for the entire department. She didn't use it. But it's fine. Okay. So all this is just a sequence of excitation. So at the first bullet point, it's going to be the SA node. Next bullet point is the AV node. Then the AV bundle. Then the bundle branches. And then the Purkinje fiber. So that's all you're writing, is that sequence of excitation that we just talked about. And that showed you exactly where they're all located. So SA node is right here at the top of the right atrium. The AV node number 2 is at the bottom of the right atrium. And now the SA node has the ability to excite the left atrium through just gap junctions. All we need is the gap junctions. We don't need an actual physical connection. And then after the AV node, then we have the AV bundle right at the beginning of that interventricular septum. And then they separate into 2 bundle branches that go to the right and left ventricles. And then the Purkinje fibers are down at the apex and up the side of the ventricles. You can see them extending up here. Those are all Purkinje fibers. Last thing we're going to talk about are ECGs, and then we'll be done.
Question?
Yeah, of course.
For number 5, when it goes up, it refers to the whole, like the long piece, right? Not just the small branches?
Yeah, so as soon as we get down to, it's all, well, it is. So it's all of these branches here. So from here and all of these guys, this is all Purkinje fibers.
Right.
And then it's all up on the left side as well. So all of this. All those are Purkinje fibers. Uh-huh. Okay, any questions about the excitation pathway? Yeah. Javen.
Can we see these on the model?
The fibers are drawn on there. Yeah, so I don't know the numbers off the top of my head, but they are like little white circles with number on them. There's one on the right heart wall. I want to say it's number 51, but I can't remember. But SA node, AV node, I'll show you. Ask me in lab, and I'll show you exactly where the SA node and AV node are in the right atrium. I'll show everyone, but remind me. Okay.
Are you going to test on this one as well?
Say it again.
Are you going to test on the models on this one?
Yep. Okay. So we're gonna go through these steps of the ECG. This is just the summary of all of them, but I'm gonna take it piece by piece. So the first wave is our P wave. And our P wave, and remember one thing I wanna show you is that this is showing you electrical activity of the entire heart together. When we're looking at an action potential, we're looking at the electrical activity of a single cell. An ECG tells us the electrical activity of the entire organ. Okay, so that's what makes this different from an action potential. An action potential is a single cell. An ECG shows us the entire organ electrical activity. And then an ECG stands for electrocardiogram or electrocardiography. Yeah. ECG is fine. Again, extra quarter of a point if you spell it all out for me. They add up. So the first one is the P wave. The P wave is showing you, and that's a wrong color to use. P wave is showing you atrial depolarization. Okay, so we're seeing the excitation of the atria first. Try to remember stuff what your notes look like here. Oh, it just shows you an ECG. Okay. So that's going to be atrial depolarization. Now the flat line in between that wave and the next wave, now there's no electrical activity happening right there necessarily, but during that flat part here, atrial contraction is occurring. You're not seeing contraction because that's physical or mechanical activity. This is only showing you electrical activity, but know that atrial contraction happens during that time. So we have atrial depolarization and the P wave, so the atria are getting excited. And then once that's complete, then the atria contract. And remember, there is an impulse delay between when the atria get excited to when the ventricles get excited. And we need that so that they contract at different times. So after that impulse delay, after the atria contract, then we have the QRS complex. The QRS complex is showing us ventricular depolarization. So what you're seeing, this QRS complex, is only ventricular depolarization. Now, during the same time, atrial repolarization is occurring. We're coming back down on that action potential, but it's not shown because it is hidden by the QRS complex. So why we only see ventricular activity and we don't see atrial activity? Ventricles are much larger and have many more cells, so that's what shows up on an ECG. Okay? And the atrial electrical activity is a smaller event, and so you just don't see it. It's masked by the ventricular depolarization. It's still happening, it's just not being read on the ECG. So for the QRS complex, it's showing ventricular depolarization, but atrial repolarization is also happening during that time. So is everybody okay with that? Okay. The next flat line between the QRS and the next wave, depolarization is now done. And during this flat wave or flat line, ventricular contraction happens. And again, that is mechanical activity, so that's not what we're seeing, but I'm just telling you that that's what's happening during that time. So P wave is atrial depolarization. During the flat line is atrial contraction. QRS complex is ventricular depolarization, and then the flat line is ventricular contraction. I had cinnamon for my coffee. Get to the back of my throat. And then our last wave is the T wave, and that is showing us ventricular repolarization. So the coming down of that action potential. And then after that, ventricular repolarization is complete, and we have some time before the next electrical activity or electrical events of the heart happen. Yeah. The faster your heart beats or heart works, the shorter that in between space is going to be. Okay, questions? All right, so now we're going to look at ECG tracings. This is just a diagram of the summary of all those waves, and that's already in your lecture notes. But this is an infant undergoing an ECG. You can see this is a 4-lead ECG. You can have like a 9-lead, 12-lead. There's many different kinds of ECGs. But this is showing you normal sinus rhythm. So this is a normal ECG tracing where you have the P wave, and I'll do it. Don't do black on black. Okay. P wave, QRS complex, T wave. P wave, QRS complex, T wave. So this is a normal sinus rhythm, a normal ECG. The next one is a junctional rhythm. So with this one, there is no P wave. All you see is the QRS complex and then a large T wave. QRS complex and then a large T wave. So you're not seeing any P wave at all. So with a junctional rhythm, the SA node is non-functional, and as a result, there are no P waves. And so when their SA node isn't functional, then the AV node has to take over. And when the AV node takes over, the heart is only going to beat at about 40 to 60 beats per minute. So you'll notice that compared to this ECG tracing, this is more spread apart. It's happening at a slower rate because the AV node can only depolarize at 50 times per minute versus the SA node at 75 times per minute. So on an exam, I could show you, or your next quiz, I could show you this tracing, and I can ask you the name of the ECG tracing. You would tell me it's a junctional rhythm. I can ask you what wave is missing. You'll tell me the P wave is missing. I could ask you what's wrong with the heart in this ECG tracing. You'll tell me that the SA node is non-functional. I can ask you how fast is the depolarization rate. You tell me it's about 40 to 60 times per minute. Or I can ask you what's the resulting heart rate, and you would tell me it's 40 to 60 beats per minute. So the numbers are going to be the same whether I ask you for depolarization rate or heart rate, but the units are going to be different. So if you hear depolarization, it's times per minute, or if it's heart rate, it's going to be beats per minute. So questions about a junctional rhythm. Okay. The next one, what's different about this one? What's missing, or what don't you see happening, or what do you see going on here?
The periods are going down.
The T wave or shorter.
Yeah. Well, the QRS complexes are shorter, and there's less of them. So what you're seeing here is here's a P wave and then nothing. But then you have a P wave, QRS complex, and a T wave like you should. Then again, a P wave and then nothing. But then you have a P wave and a QRS complex. What you're seeing here is something called, it's called a second degree heart block. So what's happening here is that you're getting the SA node working because you see that P wave, but then there's nothing after that, meaning that the AV node is not catching that signal for whatever reason. Okay, there's something wrong with the AV node. There could be something wrong with the bundle. Okay, but the signal's not getting sent down to the ventricles. So you don't see the ventricular depolarization. For some reason, the ventricles are not getting excited. So again, it's like the SA node is trying to send a signal, but it's not getting out. So then it tries again, and then yes, it catches, and then we have a regular ECG. It tries again, no signal, there's some kind of blockage, okay? But then it tries again, and it happens, okay? So with this one, the AV node fails to conduct some of the SA node impulses. So as a result, there are more P waves than QRS waves, and in this tracing, you're seeing 2 P waves for every QRS complex. It's like they knock the first time, nobody's home. But then they knock and somebody answers. Okay? So it's every other time the signal is getting sent down to the rest of the heart. Does that make sense to everyone? Okay. Any questions about a secondary heart blocks? Okay. Same questions. I can ask you the name. I won't ask you the beats per minute because that's going to be different for different people. Hey, for this one, I just will ask you, how is this different from a normal ECG tracing? You'll tell me there's more P waves than QRS complexes. And then I can ask you, what is the issue with the heart with this ECG tracing? And you're going to tell me that the AV node is failing to conduct some of the SA node impulses. Everything that's on this slide here. Okay. Our last ECG is ventricular fibrillation. This electrical activity is disorganized. Action potentials occur randomly throughout the ventricles. This results in chaotic, grossly abnormal ECG deflections or readings. And so we'll see ventricular fibrillation like this in acute heart attack or somebody who's just experienced an electric shock. So this is just chaotic ventricular activity. And this is when they may use a defibrillator to reset the heart and get it back on its normal pacing. We might go a couple minutes over, but we won't go into the next class this time. All right, any questions here? Yes?
This would be like an elevated heart rate?
It's going to be a chaotic, they're not going to have like a normal heartbeat. It's going to be everywhere, contracting like crazy. And again, this is when you would need a defibrillator to try to reset the heart, where they go, clear. Mm-hmm. Yeah, blue, yep. And this is just a summary of all those ECG tracing. These are the only ones that you're responsible for when it comes to testing, just these 4. But some of these issues can be resolved with a pacemaker. They put a device inside your chest cavity, and it could be leads or it could be wireless, but with leads, the leads are going to go out to wherever the issue is. So if it's an SA node issue, it'd be where the SA node is. If it's an AV node issue, it'd be where the AV node is, and so on. They can be as small as sitting in the palm of your hand, so a very small device. And again, can have wires or no wires. That's going to determine how long you need to have it for. Here's an x-ray showing you the placement of the pacemaker and then where the leads are. Now, one thing I want to talk about really quickly, we only got like 10 minutes before the next class comes in, but if you remember from the autonomic nervous system, we have our sympathetic nervous system that's going to help regulate the pacing of the heart in times of stress or exercise, physical activity. And one thing to know, and to put this in your notes, is that the sympathetic nervous system releases epinephrine and norepinephrine. What are epinephrine and norepinephrine called as a group? Catecholamines. So our sympathetic nervous system releases catecholamines. Cholamines. And they release them at the SA node, AV node, and the ventricles. And then the parasympathetic releases acetylcholine at only the SA and AV node, not at the ventricles. And we'll talk about why that's important on Wednesday. Now, the sympathetic over here, it's responsible for keeping our heart rate at about 75 beats per minute. If we were to cut the nerves that are attached to the heart, then the heart would still beat, but it would beat much faster. So normally the heart would want to beat at about 100 beats per minute on its own. But we have the parasympathetic nervous system, our rest and digest, to keep it down at 75 beats per minute. That's called parasympathetic tone. And you learned about that in B32. So parasympathetic tone keeps our heart rate down at 75 beats per minute. Without that connection, our heart would beat much faster at about 100 beats per minute. All right. So if you don't do that, if you need to, make sure that you go back and review autonomic nervous system. This is going to be brought up many times this semester. So look at the difference between parasympathetic and sympathetic. Parasympathetic releases acetylcholine at the very end. Sympathetic releases norepinephrine and epinephrine at the very end. And remember, it's a 2-neuron system. So make sure you review that, and we'll talk more about this when we come back on Wednesday.